Hi guys, so my name is Jessalyn. I am one of your Bio 5A SIs for Dr. Joseph. And in this video, we are going to cover Lecture 5 about proteins. If you have any questions at the end of this, my email is jyam002@ucr.edu. It is listed in blue, and my sessions are Monday 5 to 6 p.m. and Wednesday 10 to 11 a.m. So we're just going to do a quick review. We have already learned about carbohydrates. The monomer for carbohydrates are monosaccharides, and to bond two monosaccharides, you're going to form a disaccharide. You bond them using glycosidic linkages. For lipids, your monomer, or your building block in this case, because you don't technically have a monomer or polymer for lipids, is 3 fatty acids and 1 glycerol. 3 fatty acids plus 1 glycerol is 1 triglyceride, and in order to form this, you have 3 ester linkages. So 3 fatty acids are bonded to the 1 glycerol through 3 ester linkages. And today we are going to cover proteins. So the monomer for proteins are amino acids, and the bond formed between these amino acids to create a polymer or a polypeptide chain are peptide bonds. One thing I want to point out is that what do all 3 of these bonds formed have in common? They are all formed by dehydration synthesis, where 1 water molecule leaves every time this bond is formed. We are going to talk about proteins now. So what are proteins? Proteins represent 1 or more polypeptides that carry out a specific function that is dictated by their structure and their shape. So structure and shape are very important. A polypeptide is a polymer or a chain of amino acids made during the process of translation. If you look at the diagram right below it, you have DNA goes through transcription to RNA, and RNA is translated to proteins through translation. So this is what we call the central dogma, where DNA goes to proteins and they perform its function. One important thing to note is that the 3D shape of a protein determines its biological function. So we are going to go over some of the functions of proteins. Proteins are extremely diverse and they participate in a lot of different functions. And I'm just going to talk about a few right now. So enzymatic proteins are extremely important. These are just enzymes. They catalyze reactions, they speed them up. That's so important. For defensive proteins, these are things such as your antibodies, and they help prevent or they help protect you against diseases. You have storage proteins. So one example is casein, which is found in milk, and this is a protein that is beneficial to baby mammals. Another one is ovalbumin, that is found in egg whites of an egg, and that is beneficial to the developing embryo. The last one I want to talk about are transport proteins. We will definitely cover this more later on, but these are proteins that are found in the cell membrane. Other proteins you have are hormonal proteins, receptor proteins, contractile and motor proteins, and structural proteins as well. So we are going to talk about the amino acid structure. On the left side in blue is your amino group, so NH2. On the top in black is your hydrogen. On the right side in red is the carboxyl group. On the bottom is the R group, otherwise known as the side chain. This R group is a variable group, meaning it will change, and depending on what it is, it will give you the respective amino acid name. I also want to say that within the human body, there are only 20 amino acids that are used to build proteins. In order to form a protein or a polypeptide, the peptide bond is formed from dehydration synthesis. So on the left, you have one amino acid, you have a second amino acid, the OH from your carboxyl, and a hydrogen from your amino are bonded together and leaves as H2O, creating a carbon-nitrogen bond. So this is your peptide bond. Properties of the R groups. So your R groups, otherwise known as side chains, are variable groups, and they give the amino acid its specific name. The first one I want to point out is cysteine. This is actually a type of amino acid, and this is the only one that contains sulfur in its side chain. Thus, it can create a disulfide bond later, and we will talk about that when we're covering the structure of proteins. The other properties include hydrophobic and hydrophilic. So if you have a hydrophobic R group, that means the side chain is nonpolar. If you have a hydrophilic side group or R group, then the side chain is polar or electrically charged. And what do we mean when we say electrically charged? Electrically charged side chains are either acidic or basic. The first thing I want to cover of the structures of proteins is primary structure. So I'm actually going to pull up a document right now. Okay, so primary structures is composed of a sequence of amino acids. So this is just a polypeptide. Each of these little circles represent one amino acid. This is the structure within each amino acid, and then the three-letter names are acronyms, and these are not, they're abbreviations for each different type of amino acid. Between that, you see is a peptide bond. The primary structure is responsible for dictating the size of the protein and the identity of the side chain along its length. Okay, so moving on to secondary structure. Secondary structure is where hydrogen bonds can occur between the oxygen of the C double bond O and the hydrogen in the NH. So as you can see here, this is your hydrogen bond, and you have your NH and your C double bond O. Interactions in the secondary structure only occur within the backbone, and you can form two different shapes. So you have the alpha helix, so down here, or you have the beta pleated sheet. Next we're going to talk about is our tertiary structure. So this is our polypeptide backbone, and in our tertiary structure, this is where interactions occur between the amino acid side chains, or their R groups. There are four possible interactions, so we're going to go through them. Number one, hydrogen bonds. So electronegative O and the hydrogen, that's a hydrogen bond right there. You have hydrophobic interactions. So these side chains are both nonpolar, and so that's why we call them hydrophobic interactions, and they are stabilized by van der Waals interactions. The disulfide bridge here is created by the two cysteine amino acids. So if there's one cysteine here, another cysteine here, the sulfurs bond together and form a disulfide bridge. And the last one I want to point out is ionic bond. This is also called electrostatic interaction, but we know it's an ionic bond because they are both charged. So the NH3 has a positive charge and the oxygen has a negative charge. And for the last one, there is a quaternary structure. So these consist of multiple polypeptides. This is collagen. You see that there are multiple chains together. And over here is what we're going to focus on. This is hemoglobin. There are 4 different polypeptides here, 2 alpha helixes and 2 beta subunits. And this is our overall structure. So you see 4 polypeptides together. Okay, So remember what I said is important. If you change the shape, you'll alter the function. So if we change the 3D shape of a protein, it will alter the function, and sometimes it could be detrimental. The two ways that we learned of altering 3D shape is one by denaturation, where this is an environmental effect. So you can affect the shape of a protein by like high temperature or changes in pH, whether it's too acidic or too basic. This can lead to the denaturation of a protein. The other one that I want to talk about is mutation. A mutation, for example, sickle cell anemia, where red blood cells are deformed and don't have their normal circular shape, is because of a genetic mutation within its DNA. Okay, so this is the end of the quick review on proteins. If you guys have any questions, you guys can leave in the comments below because we will be responding to those. If not, send me an email or you can also come to my essay sessions. Okay, thank you.